DNA is a biopolymer made from small building blocks known as bases. Complementary bases have a propensity to form hydrogen bonds between two single strands of DNA, known as Watson-Crick base pairing. Adenine pairs with thymine, and cytosine pairs with guanine to create a stabilized double helix structure. Nadrian Ned Seaman first proposed using DNA as a means for building nanostructures in 1982. Modern methods of DNA origami engineering were then developed by Paul Rothmund. It is interesting to note that both of these scientists published this research as single-author papers, which isn't seen often. DNA origami is a branch of nanotechnology which uses long, single strands of DNA as a scaffold to engineer a variety of two-dimensional and three-dimensional structures. The scaffold DNA is often derived from the 7.2 kilo base pair genome of the M13 bacteriophage. This construction can be accomplished by designing shorter single strands of oligonucleotides called staples. Staples are typically 20 to 70 base pairs in length and are designed in such a way to be complementary to different regions within the sequence of the scaffold strand. They form crossovers or crosslinks between distant regions of the scaffold strand, bringing them together and creating a fold. DNA origami synthesis can be performed in a single reaction. First, the scaffold DNA strand and hundreds of staples are combined. The mixture is then heated to separate all the strands, which then self-assemble into the desired nanostructure upon cooling. The newly synthesized nanostructures can then be visualized using different techniques. These include atomic force microscopy and electron microscopy. Some of the first two-dimensional structures created by Rothman were imaged using atomic force microscopy and included a square, a star, and a smiley face. In the same study, patterned origami was also investigated and produced intricate designs such as lettering and a map of the Western Hemisphere. More recently, DNA origami has been used to engineer and synthesize a wide range of different three-dimensional nanostructures for a variety of purposes. Three-dimensional DNA nanostructures offer one way of improving the performance of biosensors, with implications in the diagnosis of health conditions as well as monitoring molecules in the environment. For instance, nanomachines are nanorobots that possess the ability to change the molecular conformation in the form of some type of movement or action in response to a stimulus from their environment. The idea of encapsulating molecules has made three-dimensional DNA origami nanostructures highly appealing to researchers. The nanostructure would help to preserve bioactive molecules while traveling around the human body until they reached their intended area of effect. Douglas, Bachelet, and Church used DNA origami to create a cage-like nanorobot that is regulated by an aptamer-encoded logic gate for the targeted delivery of drugs and other materials such as nanoparticles. Several AND logic gates were also engineered within the structure, such that its opening and delivery of whatever material it was carrying, referred to as its payload, would only occur when specific signal molecules were released by nearby targeted cells. Zhang and his colleagues showed that DNA origami nanotriangles were highly effective in delivering a known chemotherapy drug, doxorubicin, into breast tumor cells in mice without toxicity and a reduced risk of side effects. Overall, DNA origami has become a very useful tool for the bottom-up synthesis of DNA nanostructures from rational design. One challenge that researchers continue to face is maintaining the stability of the DNA structure in a range of physiological environments. Although this field of research continues to advance rapidly, it would greatly benefit from future endeavors which generate better and more accurate models for individual origami structures, as well as potential larger assemblies made from several origami structures.